Okay, so we went through some of that stuff kind of quickly. Unfortunately, I'm not going to be here to answer some of your questions, but I didn't want us to lose any time. So I threw this together real quick to see if we could maybe answer some of your questions and give you a better chance to work on some of your practice stuff in class today. So I wanted to hit on a few things first before we got into the genetics of the blood typing and how to figure out if mom is one and dad is another, what are the kids going to be like, and stuff like that. So first, when we're talking about blood and blood type, we have to talk about the antigens and the antibodies. Antigens are the ID cards, they're the pass. They are what tell the rest of your body that you are supposed to be here. Okay, the antibodies are the hall monitors. Somebody in another class had that, and that was a really good example. They are roaming the halls, aka your blood vessels, and they're checking to make sure that everybody's who they're supposed to be. And if they find somebody who's not supposed to be, they're going to grab onto it, and they're going to clump together, and that's that process known as agglutination. Okay, so if I have A antigens, I'm going to have a certain shape to them, the same thing with B. If I have AB, if you notice, they have both. O has none of them, all right? But they produce both sets of antibodies. When we're looking at donating and receiving, one thing to keep in mind is you can always donate and receive from your same blood type, okay? So if I'm A, I can give to A, I can receive from A, okay? Now, some special circumstances, we got something called the universal recipient. So if you notice right here, AB can receive blood from anybody because they don't produce any antibodies to reject those new blood cells. But they can only give to other AB, all right? They can't give to anybody else. And O is opposite. They can only receive from O because they have all of the antibodies. So if they take in anything that is not O, those antibodies are going to destroy those cells. They can, however, give to everybody. And so that's why they're known as the universal donor. Okay, so that's kind of how that works. RH factor, that's your positive and negative. And basically the big takeaway with that one is if you are RH positive, so we're saying you are A positive, B positive, O positive, that sort of thing. Okay? If you are positive, you can receive from positive or negative. Okay? If you are negative, so like A negative, you can only receive from negative. So if you're positive, you can receive from everybody. If you're negative, you can only receive from negative. And there's other factors that medical professionals use when they're doing blood typing and other protein markers and stuff like that. But we just need to look at the A, B, and the RH factor. So basically when we're trying to figure out what the kids are going to be, we want to look at the alleles. Now think back to your biology class. So, you know, what did Mr. Munich teach you about this stuff? We've got alleles. Alleles are different versions, okay? We've got dominant and recessive. That's what you were probably used to when we first went over this stuff. This is a little bit of a different scenario. It's a multiple allele scenario. It's also used in some circles. It's a codominant circle allele sort of thing. But basically how it works, if you're going to have a particular blood type, you have to display the antigens. So you have to have those particular alleles. So for example, if you're going to be type A, you can either have AA, because remember you're getting some alleles from mom, you're getting some alleles from dad. So mom can give you A, and dad can give you A, and that gives you type A blood. And that would be the, dominant condition, okay? Or you can have mom can give you A and dad can give you O, but you still have A because O would be considered the recessive. And what we know from genetics, we know that dominants cover or mask recessive. So you're not going to see that recessive allele present. It's going to be there, it's just going to be hiding it, okay? or it's going to be hidden by the dominant phenotype, the physical expression. And if they're different, A and O, they are heterozygous. Hetero meaning different, right? Same thing with B. You can either be big B, BB, homozygous dominant. Hopefully these words are starting to ring a bell a little bit. Or you can have at least one B and an O. Here's where the codominant part starts to come in. If you are AB, you have A and you have B. There is equal expression. So in order to have AB blood, you have to have A and B. O is the homozygous recessive, so that means we have to have two O's. Now, sometimes you may see them referred to as I's, and then you'll see them as I, A with big I's, and B like that. We're going to stick with the letter A, the letter B, and the letter O. I find that makes things a little bit easier. And if you're interested in seeing how it works, I can show you the big I, little I thing if you want to. So basically, this is how it's going to work in practice. We can use a Punnett square. That's the 4-square thing. It looked like that. Most everybody got pretty excited with the 4 squares because they could get those pretty easily. So basically, what we're going to do is use a Punnett square to figure out what the potential offspring are going to be. So if I've got a type AB female and a type O male, so we've got our Punnett square, we've got our 4 squares, and we're going to fill the boxes. Around the outside, we're going to put the alleles of the parents. So on one side, I'm going to put mom. It doesn't matter what side you put them on. The results inside the box are going to come out the same. Remember, if dad is type O, the only way to have that is if I have 2 O's. I'm going to bring my columns down like this, and then I'm going to bring my rows across like this. And so the possible blood types are the offspring. I've got AO, which means it's type A, here and here, and then here, if I've got a BO, that is B. So I've got 2 there. So that means 50% of my offspring can be type A, and 50% of my offspring, or at least the probability of them being B, is 50%. And that's how that works. So basically, when you're doing the Punnett squares, you want to figure out your parents, write them around the outside, then fill the boxes in. I would recommend, let me go back, I would keep this handy in front of you. You should also have it in your notes, and if you don't, I would write that down. Keep that with you. Have that so you have the list of the possible genotypes, and then eventually, as you do enough practice, you'll probably get it. If you have any questions, you can hit me up on Remind or send me an email message, and I'll try to get back to you as soon as I can. If not, we can talk about it briefly when I see y'all again on Monday. Don't forget to do your notes on the forensics of blood and other body fluids, and then there's some homework that you should have picked up to work on also. Good luck, everyone, and I will see you later.